Assalamu alaikum. Dear student, today we will discuss about the second lecture of genetics. This topic is very important and this is Human Genome Project. In this lecture, we will discuss what is Human Genome Project, what is the purpose of Human Genome Project and how many years this project took to complete and what are its medical and economical importance of this project. Human Genome Project is actually it is the world's largest collaborative biological project. The idea was given by US government and it was given by US government in 1984. But this project was formally launched in 1990 and the whole project was completed in 2003. The main objective of this project is to sequence the nitrogenous bases of that gene that control the character, that control the trait of humans. Because here we are discussing about the Human Genome Project, so we will discuss about the genome sequence of humans only. But always keep in mind all the organism, all the living organisms, their characters are controlled by genes. So, when the Human Genome Project was designed and when the Human Genome Project was completed, it was not based on one individual, but this genome is a mosaic genome that represent the whole humans, not one particular individual. There are different goals of Human Genome Project. Actually, approximately in whole body of a human, there are approximately 20,000 to 25,000 genes are present. So the aim of Human Genome Project was to identify and map all these genes. The second aim was, as the nitrogenous bases are in human is about 3 billion chemical bases are present that make up the DNA. As you know, the gene is the part of the DNA and gene is made up of nitrogenous bases and DNA is also made up of nitrogenous bases. And approximately in one human, there are about 3 billion nitrogenous bases. And the aim of Human Genome Project was to sequence of that 3 billion nitrogenous bases. What is the sequence? Either, as you know, there are 4 nitrogenous bases actually: adenine, guanine, thymine, and cytosine. Sequence mean to determine the arrangement of that genes. Then third goal was to when we will map the genes and when we will determine the sequence of that particular genes, we need to store these informations in database, so in future we can easily found these sequence and we can recognize that particular disease or whatever we want to do with these genes. Then this goal is also was to discover more efficient technologies for data analysis, and this project allow the private sector access to information and technologies that arise from this project. And Human Genome Project also sequence the genome of other organism rather than humans, that include mice and Drosophila as well. And the most important thing is to use the Human Genome Project to address ethical, legal, and social issues as well. There are different agencies that participate in Human Genome Project. As we have discussed, it is the world largest collaborative biological project. So in 1990, when it was formally launched, two major funding agencies contributed to complete this project. Maybe without the help of these agencies, the scientists unable to perform this task and unable to complete this project. These two funding agencies are actually US Department of Energy and the second one is National Institute of Health. They developed an MOU and where they discussed about the plan, where how we can complete the human genome project, and they also set the timing, how in which time and how many years this project can be taken. All that things was discussed in that meeting. Then this project was performed in 20 universities and research centers in United States, in United Kingdom, Japan, France, Germany, Canada, and China. With these 20 universities and research centers, this project was also performed side by side in Celera Corporation, the Celera Genomics, which was formally launched in 1998. Because in science, when the result by one scientist was not accepted, rather than the same experiment was performed many scientists and the result was the same. That's why 20 universities and Celera Corporation performed the same thing and the result was the same. Then the most important thing is economic. So about $3 billion was formally launched in 1990 to start this project, and $3 billion was given by U.S. Department of Energy and National Institute of Health. Human Genome Project was actually a 13-year long, because it was started in 1990 and it was completed in 2003. Publicly funded project that was initiated in 1990, as we have discussed before, and it was completed in 2003. As you know, in one cell of a human, there are 46 chromosomes present. So each chromosomes have different genes, and different genes control different characters in different organisms. So in 46 chromosomes, you can say there are 23 pairs of chromosomes, and first chromosome is the largest chromosome, and all the 22 pairs was completed in 1990, but the first chromosome pair of first chromosome was completed later. So the whole project was completed in 2003. Then there are a few scientists who was the pioneers in human genome project. The first one who gave the idea of human genome project was Robert Schenchmeer in 1985. Then there are two scientists who proposed the budget of human genome project was Charles Delisle and David Smith. Then Human Genome Project was the act of Human Genome Project was passed in U.S. conference under President Reagan in 1988. Then James Watson, who was the head of National Institute of Health. Actually, James Watson was as a head of this project. Then Franz Collin, he was succeeded James Watson in 1993 as the overall project head and director of National Institute of Health, and which later became the National Human Genome Research Institute, which is also called NHGR1, and was in power until the completion of Human Genome Project in 2003. Sorry. Then other scientist who was the PhD scholar in the University of California, he developed a software, gig assembler, that allowed the publicly funded Human Genome Project to assemble and publish the human genome sequence. Then here timeline thoroughly by year when the Human Genome Project was completed in stepwise process in 1970. Before the idea of Human Genome Project, which was given in 1985, but before that idea in 1970, a scientist, Frederick Sanger, you are much familiar with this scientist, developed a technique for DNA sequencing. And DNA sequencing, it means we want to know about the nitrogenous base sequence of a particular gene. And he performed the DNA sequencing through a method which is called Sanger method, with the name of a scientist. In 1985, other scientist Robert at UCSC, he proposed the idea of sequencing of human genome. Then in 1986, US Department of Energy and National Institute of Health came forward to fund Human Genome Project. In 1985, United Kingdom Medical Research Council joined the Human Genome Project. Then in 1980, Human Genome Project was officially launched with James Watson as its project director. James Watson, you are familiar with the scientist because he proposed, Watson and Crick both proposed the model of a DNA, which is double helical DNA. A Human Genome Project was directed by Watson, and in 1990, with this, the first gene was mapped, and that gene was BRCA1 gene, and this gene code for breast cancer. In 1993, first five-year plan was published, and Sanger Institute of United Kingdom joined Human Genome Project. In 1994, Human Genome Project, the mapping of Human Genome Project was achieved, and in 1995, Genetic Privacy Act was passed, and first bacterial genome was sequenced, and that bacteria was Haemophilus influenza, and this is very important. Actually, you should know about this. In 1996, first human genome gene map was published, and with this, the yeast genome was also sequenced, and the human genome project, with this, the genetic mapping of mice was also achieved. In 1997, National Institute of Health became an NHGR1, and the genome of E. coli was sequenced. In 1998, second five-year plan of Human Genome Project was published, and in 1999, the sequence of human chromosome 22 was completed and was published in a journal, The Nature. In 2000, working draft of human genome completed. US President and UK PM support free access to human genome information, and genome of Drosophila melanogaster and Thaliana, which is an algae, was sequenced, and it was also published in journal The Nature. In 2001, working draft of Human Genome Project was published in journal The Nature and Science. In 2002, working draft of mice genome sequence was completed and published, and in 2003, finished version of Human Genome Project was completed, and Human Genome Project ended with all the goals achieved. Then there are technical aspects in Human Genome Project. The process of determining the human genome first involved genome mapping. It means characterizing the chromosome, and mapping the genome is also called genetic map. The next step is DNA sequencing. It means to determine the order of DNA bases on a chromosome, and this is also called physical maps. Mapping strategies, there are different ways through which we can do genetic strategies, are actually genetic markers we can use. And what is marker? Marker is actually any hereditary physical or molecular characteristic that are different among individuals of a population. And genetic map shows the relative locations of these specific markers on the chromosome, and the examples are given here is restriction fragment length polymorphism, in which the restriction enzymes are used, and these enzymes recognize the short sequence of a DNA and cut that specific sites. Therefore, DNA can be cut into many different fragments, and these fragments are DNA pieces used in physical maps. Restriction fragment length polymorphism actually reflect the sequence differences in DNA site which are cleaved by restriction enzyme. Then a sequence strategy. Before sequence the DNA, it must be amplified or increase the quantity. There are two different methods through which the quantity or amplification could be done. The first one is through cloning, and the second one is through PCR. In cloning and PCR, if we have one copy of that particular gene, we can make thousands of copy of that particular gene. And the sequence technique could be done through shotgun sequencing method, or it could be done through Sanger sequencing method. Here is a shotgun method is shown. Here in red, you can see there are many copies of gene, and we insert that gene in bacteria where multiple copies could be done. It could be sequenced, assembled, and particular sequence of gene will be achieved. The second method is Sanger method, and in Sanger method, as you know, there are four nitrogenous bases. We need to take four test tubes, and we will label all these test tubes with guanine, thymine, adenine, and cytosine. And in each test tube, we need to add all the important ingredients, all the important bases, and ingredients mean we need to add primer, we need to add enzymes, and with these we also need to add one particular important thing, which is dd and tp. For example, in guanine flask we need to add dd and gtp, and in thymine flask we need to add dd and tp, and in adenine we need to add dd atp, and in cytosine flask we need to add dd ctp. Then after adding this, we need agarose gel, and the sequence will be done, and the sequence will be stopped where the dd atp or ddctp will become, because that ttp gtp atp and ctp will stop the sequencing further of that particular gene, and by doing this we will get our particular sequence of that gene. Then there are outcomes of human genome project that we have discussed in before. In 2003, at the end of 2003, we achieved all the aims that we that was concerned with human genome project. As we have discussed, there are approximately 22,000 are many genes are present in humans in one done, and with this the sequence of mice gene was also done, and Drosophila and C. elegans, which is the earthworm, all the genome sequence was done at the end of 20. A large proportion of our genome that encode a transcription factor is dedicated to control element to which these transcription factor bind, and the combinatorial use of these elements provide much greater flexibility of gene expression than found in Drosophila and C. elegans. So the major outcome was also to determine the proteomics of human genome, and it is also concerned with the other organisms like Drosophila and roundworm. Then gene density. On chromosome number 19, there are about 23 genes, and 23 genes are coded with 23 genes per million base pairs on chromosome number 19, and on chromosome number 13 there are about 5 genes per million base are antibodies and T cell receptors for antigen, and transplantation antigen of major histocompatibility complex, which is also called MHC, and human leukocyte antigen, which is also called HLA. Cell signaling molecule, including the many types of cytokines, the molecule that participate in blood clotting. Human genome comprises of 2% of exons and 98% of enteron, and exons are actually coding regions and enterons are actually include non-coding regions. There are few applications of human genome project, and you know what is the purpose of doing human genome project. Actually, the major application and the major usage of human genome project is we can use it in molecular medicine to human evolution. It also help to identify disease causing gene. It also help to identify the mutations. It also help to sequence of a DNA which is stored in database. And the US National Center for Biotechnology Information house the gene sequence in a database known as Gene Bank, along with a sequence of known and hypothetical genes and protein. And human genome project also allow for advance in agriculture through specific gene modification, and it also help in forensic science as well. I hope today's lecture is helpful for you. If you like the video, please like, share, and subscribe the channel. Stay connected. Allah Hafiz.